Welcome everybody to the intro to the microbiome. I am Francisco Rodriguez with Supplement Innovator. You are looking at the global headquarters, which is a fantastic office in West Palm Beach, Florida, and we are excited to share with you the first webinar that we are going to do of a few webinars on the microbiome. I want to tell you a little bit about Supplement Innovator briefly. Supplement Innovator is a consulting firm that works with brands that are looking to launch new products, as well as raw material supplier sales teams, provides training and coaching to those sales teams. And we have one area that we like to cover under special projects, and that is where we do some things that we think are not only exciting for the industry, but also important. And they are on the cutting edge of what the industry is going to be doing, what the industry is able to participate in, what can benefit the industry. And this webinar and the series of webinars on the microbiome, I believe, will provide that next level of innovation. We are being joined by someone that I want to introduce, but before we do that, I want to introduce his company. Second Genome is a next-generation sequencing company, and they're focused on microbiome science for discovery and development of therapeutic products. The company has established a pipeline of microbiome modulators that impact infection, immunity, and metabolic diseases. Second Genome's development pipeline is funneled by novel technologies for identifying, screening, and scientifically validating product candidates and microbial biomarkers. Second Genome's technologies have been rigorously validated through partnerships with leading pharmaceutical companies and nutrition companies, as well as academic and governmental research institutions. Some of the recent activities that are really newsworthy: Second Genome advances lead microbiome drug delivery development program in phase 1 clinical trials as a treatment for inflammatory bowel disease, and they are working in collaboration with the Mayo Clinic on a variety of diseases, and we are glad they are joining us for today's call. Specifically, we are going to be joined by Take Ogawa from Second Genome. Take is the Director of Research Services at Second Genome. He has spent the last 15 years in the life sciences industry developing and promoting genomics tools and applications. Take was part of the development team that launched the first high-throughput sequencer at Applied Biosystems that was responsible for sequencing the first human genome. He also promoted the first next-generation sequencing platform at 454 Roche. According to Take, he has seen the industry shift the research focus from looking at just changes in our human genome to also including at how we interact with the environment around us. Ladies and gentlemen, I am delighted to welcome Take Ogawa to the interview and the presentation he will be presenting to us. Take, thank you for joining us.
So thank you for having us, Francisco. Appreciate it.
No problem at all. And Take, I'm going to share the presentation that I know that you are excited to present to the group, and we can go ahead and get started.
Absolutely. Great. So as Francisco mentioned, we'd like to take this first webinar, first of several webinar in a series, to first, of course, introduce the concept of the microbiome, the concept and really define the microbiome. Why is it important to us? Where did it come from? Why is it important to us? And where is this all going? And so I want to just take a few minutes of your time here to get everybody on the same page as to how we define the microbiome today, and go through some examples, some real anecdotes about how valuable the microbiome has become for us in our academic and also industrial applications in research. Next slide. So the term microbiome is actually relatively new. It was coined back in 2000, right around 2000, 2001, by a group that included Joshua Lindenberg, who coined this term, the microbiome, to define the ecological communities, the ecological systems of microbial communities. So these are microorganisms that include, of course, bacteria, archaea, yeast, and viruses and such that live in a symbiotic fashion oftentimes, and they literally take up residence again in a symbiotic fashion within us and upon us in the various parts of our body. And so if we go to the next slide, here's just again a simple cartoon, a simple depiction of what we have discovered thus far about these communities. And so again, they literally reside in vast numbers and vast diversity in various parts of our bodies. And so as again depicted here, we see that various organisms reside in our mouth, various organisms reside on our skin, and of course a major community is in our GI system. And so to take away from the idea that all bugs, all microbes, all bacteria is bad, in fact a lot of these organisms are absolutely essential to our well-being. So the natural question comes then, where do these bugs come from? Where do these organisms come from? And so for a long time we have believed that humans are born germ-free, that when we come from our mothers, that the bacteria that she has on her body, in her skin and in her vaginal canal and such, in terms of a natural birth, is where we are basically inoculated, if you will, by these bugs and these organisms. Turns out, though, in recent studies, and by recent I mean just in the last year or so, we've discovered, in fact, that we are not 100% germ-free, even during gestation. And so there's been several microbiome studies to show that, in fact, there are microbial communities. The microbiome actually resides in the placenta and various other parts during the gestation process. So the science is definitely evolving in terms of how and where and why these bugs take us upon as hosts. And in those studies, we've also found that, of course, these communities are very dynamic. That once they colonize us in our bodies, that they in fact are constantly changing. That there's been studies that show that in the first 2 to 3 years of our lives as infants and toddlers, that the microbiome is very dynamic, and that after those couple of years that it starts to settle. And of course there's correlations between, is it a switch from breast milk to solid foods, is it just the time taking its course, or whatnot. So there's a lot of hypotheses about that, but to that point, again, these microbes that reside within us and upon us are very dynamic. They're constantly shifting. Literally, they are, of course, live organisms, and so they're growing, they're coming and going, and they're in competition with one another. And of course, the environmental aspects in terms of, again, what we eat, drugs that we may take, antibiotics, environmental exposure, stress. All of these things affect which organisms might be colonizing at any given point in time. And in these studies, again, in the last decade or so here, we have discovered that there are in fact hundreds, if not thousands, of species of bacteria and organisms that reside and take us up as a host, particularly in the gut. And also in, again, various other places within our bodies. But it happens that our gut microbiota, microbiota being defined as the actual community of these organisms, is the most diverse of any location within our bodies. And then the study of that microbiota, again, is what we define as the microbiome. And what's really interesting here as we start to philosophize and hypothesize as to why, well, why do we need these bugs and why do they need us? And you could argue, of course, that, well, the bacteria need us because we feed them. We literally provide a comfortable home and food and shelter, if you will, for them to nurture. But what do we get out of it? And as shown here, it turns out these organisms as a collective entity, a collective community, is represented of millions of genes. These are functional elements of their DNA where they are generating, producing various metabolites, proteins, and so on, various bioactives that are at interplay within our bodies relative to our own DNA within our own cells, which only consists of about a couple dozen thousands, a few thousand genes themselves, where again these microbial communities consist of orders of magnitude more genes. So you can say that they are playing, we do actually know that they play an essential role in our immune system and various other pathways and functions within our body to keep us healthy and functional. And so as these organisms go into dysbiosis, that is an unbalanced state where organisms of a pathogenic nature are overly represented, that's where our function in our well-being starts to fall apart. And so again, they play an essential role in our maintaining ourselves. You could argue that it's the most dynamic part of our biology, hence the concept, the second genome, relative to our own genomes. So as we study these microbial communities within the realm of our own bodies, the human body that is, we find that again the makeup of these communities vary quite significantly depending on the location within the body, the gender, of course, and age again can make a significant impact into these communities as well. So what we're seeing here is that each pie chart represents a different sample within a different part of our body, and each color represents a different group, a different type of bacteria that resides there. So you can see not only are there different types, but the representation, the ratios of the overall community in terms of the way they're represented within a community varies quite significantly from one location on the body to the next. So as we start to decipher the complexity and the interplay of these microbial communities within the context of our human bodies, we are finding through various academic and research endeavors that, in fact, the microbiome correlates and was associated with various disease states or health states, for that matter. So here's just a subset of examples of various disorders and diseases that have already been shown to be associated with changes in our microbial community. So, of course, microbial communities do differ between individuals, depending on, again, all the various variables that we discussed, but it also turns out that individuals within a certain disease state, so for example, obese individuals have a very unique microbial community signature amongst themselves relative to quote-unquote healthy individuals that we would not consider obese. And so we're seeing, again, strong association between the types of bacteria, the types of microbes that reside within these disease states. And again, to really take home the idea of the increased level of interest and thus the science and the findings that we're finding, it's really daunting to think that basically in the last decade here, the amount of knowledge that we've gained, and this is basically depicting the number of peer-reviewed journal articles that have been published. So these are research studies that have been published on the topic of the microbiome. So this can really range beyond just human health into many other applications. But basically, most of this is in the realm of human health. in human disease studies. And again, you can see that the number of the knowledge is growing dramatically. And I should also point out at this point that when we look at this timeline, and we can start to beg the question of, well, why did this start to explode in 2005, the time frame? Because we've known about the idea that bacteria, for better or for worse, have been in our environment, have infected us, have inoculated us, have resided on us and in us since the dawn of time. We've known this for a very long time. But why has there been this explosion in the science of the microbiome? Because again, the microbiology as a scientific discipline has been around for a very long time. The reason really is two things, two factors. One is the advent of technology to look at genomics. So genomics is, of course, the study of DNA, really, and genetics on an overall scale. And the advent of technologies that allow us to sequence the DNA, to measure the DNA, to analyze and interpret the DNA, really started around this time frame of about 2005, between 2000 and 2005. So only with the advent of those sort of technologies have we been able to look at these bugs, these organisms, at the DNA level, which of course allows us to identify these organisms with much more granularity and resolution. But of course, with the advent of these sort of technologies, we have far more data. And as we all know, with again, the advances in computer science, data science, cloud computing, really goes hand in hand with the more data we generate, the more we can analyze that data. So really with those two major pieces, which really converged around 2004, 2005, and sort of that time frame, we saw an explosion in this field in terms of, again, the insights we have gained in these microbial communities that reside within us. So really, what is the end game here? As we can all start to surmise, is it's one thing to identify which organisms are in these communities. That's part of it. But really, what we're trying to understand here at Second Genome, and really all of our collaborators and other groups that research the microbiome, is that what is the functional impact of these organisms to us in terms of maintaining our health, supporting our immune system, supporting our metabolic systems, preventing disease or causing disease. Really, we want to understand that mechanism of these microbial communities based on the changes that we see in these communities. Does that correlate? Does that go beyond correlation association to some causation and mechanism? So really, that's the frontier of the science today, is again not going beyond just identifying the kinds of organisms that might be beneficial to us, but what are they doing? What are they doing to us, and what are we doing to them? That's the leading edge of the science today, and that is what we are working on. So I want to share this very, very cool, for the lack of a better term, a very cool study that came out of the Washington University, out of Jeff Gordon's lab, which really highlights the true function, the true mechanism of these microbial communities. And so what they did in this study was they took the microbial community that resides within the gut. So they basically took a fecal sample from their stool from sets of twins. Now these twins were unique in the sense that they were identical twins, genetically identical in their own DNA, but one of the pair of the twins was obese and the other was considered lean. And they took samples of their fecal material and transplanted that material into model mice. And what they found, that even though these mice were also genetically identical and they were also on the same kind of diet, a low-fat, high-fiber diet, that the recipient mice that was transplanted with the microbiome from an obese individual actually also became obese themselves. And so this shows direct impact that with all things set equal, including their own DNA, that simply by transplanting a microbial community into these organisms, it actually changed their biology, that it changed their clinical condition. And to support this hypothesis even further, what they did was they caged these animals together. So what's highlighted in the bottom figure there, which are the pink animals, are the animals with that have the obese microbiome implanted in them, and then the blue animals are the animals with the lean microbiome in them. When you house them together, it turns out, of course, that they start to share the microbiome because they're living together. Their stool samples are through the environment. They're basically co-sharing starting to co-share the microbiome. And it turns out that the lean animals continue to be lean. So there's none of the obese microbiome being inoculating the lean animal, while the lean animal had by being co-housed with the obese animal actually made them leaner. So just by co-housing these animals together, they were able to have a protective nature on the animals that were obese. So that's an interesting study. Now, if we took the same situation but switched their diets to a high-fat, low-fiber diet, we found that the lean animals did not have a protective function on the obese animals, and thus the obese animals continue to go obese. So really showing that it is a complicated model of not just having a quote-unquote good microbiome, but really the environment in terms of the diet, of course, and other environmental factors in terms of who do you reside with, what kind of microbiome do they have, also affects your clinical outcome on the back end. So really highlighted the true mechanistic nature of the microbiome. So now that we know the microbiome is important to us, well, why aren't we doing more of this? Why aren't we looking at it? Why aren't more people looking at this? Well, it turns out that the microbiome, as you can again imagine, is very complicated. It is complicated in its makeup, complicated that it is literally a living, breathing thing, and thus it is constantly changing. And therefore, it's really hard to decipher. It's really hard to analyze. And so when we look at these sort of complicated puzzles, we need the right tools, we need the right expertise to put this complicated puzzle together and put it into context that we can start to answer these hypotheses that we might have. So needless to say, the more important things are in the world, of course, they tend to be harder and harder to make sense of and to decipher. So just to make that point is that this is why why groups like us have dedicated 100% of our efforts to decipher and elucidate the microbiome. So in broad strokes, what are the components of doing a microbiome study? What are the components that really can affect your insights and know-how, or what are the know-hows that really allow you to gain the deepest, the most accurate insights in terms of understanding the microbiome in the context of the questions that you have in hand? And it turns out, again, as you can imagine, that sample preparation, and what I mean by this in broad terms is what are the right samples to collect, depending on the questions that you want to ask. Is it stool samples? Is it skin samples? Is it oral samples? What is it? And where is it being collected from? How is it being collected? All of these factors can really affect the insights that you gain on the back end, either in a positive way or a negative way. And then assuming that we do address those questions and answer those questions, well then, what about how we measure the microbial community? How do we measure the microbiome such that we're accurately measuring what's really there, and we're not introducing any sort of biases in the data that we are trying to generate as we try to analyze these microbial communities? So how do we measure? What are the right technologies that we use? This is a big question that needs to be answered. And then finally, when we do generate those measurements, well, how do we make sense of that data? How do we make sense of those measurements? And that's what we call bioinformatics, which is, again, as I alluded to earlier, is the intersect of of computer science and biology. So looking at biological data, using data science and computer science to try to interpret that vast amount of laws and detect that signal above the noise to gain that deeper insight into how is the microbiome at play in the context of the questions that you have about biology. So I'll stop there, and I'll let Francisco take over here in terms of future webinar topics as we dive deeper and deeper into these subject matters.
Yeah, it's important that we not just focus on the introduction, because for the supplement industry and the nutrition industry as a whole, I think we need to look at being very targeted in what we do. So what are the benefits of the microbiome to the nutrition and supplement industries? Many people automatically move in discussions that I heard at NBJ and around the industry, they focus on probiotics in the microbiome. So I thought it would be important for us to look into that very deeply. And then what about going beyond probiotics? When we look at things like, of course, fibers, prebiotics, but what about proteins, diet and exercise, and polyphenols, different components that we introduce as an industry to the biology of the individual, but also to the biology of the microbiome. How does that cause a shift? And then what does that shift mean? And then if you wanted to start your own microbiome research for your products on the finished goods side or even on the raw material side, what would that look like? What are some of the recommendations? And Second Genome, the reason we've brought them in to present these pieces of this webinar and future webinars is because they are the experts in the industry, focusing on the commercialization of concepts. And I think the nutrition and supplement industry have a lot to benefit from those capabilities. So we want to take these last few minutes and give an opportunity to people in the community that have been asking questions, and give them a chance to ask some questions about what you've heard today. And if you've been on the YouTube channel, you can go over there and glance at some questions or comments, or if you want to shoot an email to Tracy, Tracey at supplementinnovator.com. You're welcome to shoot those. I have a few questions for Take that maybe he can answer for us regarding the introduction to the microbiome. Take, you mentioned that there are some correlation around the microbiome shift. Can you give some examples of diet correlations, or do you have some examples? And I think the mouse one is a great one, maybe on the human side where interventions were given and there was a shift in the microbiome, or, and maybe you'll give a better example, it prohibited a shift from happening from a different intervention. Can you talk about that?
Right, absolutely. And so as I hinted and as you suggested as well, for the most part, our studies in the findings from our studies have been relatively limited to association. So that is that microbial communities seem to be different in individuals or groups of individuals with a certain condition, a certain disease state. Or not even a disease state, but again, it could be that they're on a certain type of probiotic or a certain type of supplement. That groups of an individual under a same treatment could have a very different community than individuals that are not on that treatment or are not of that disease state. So that is true, but in the end, currently our knowledge is relatively limited to just association, because that is just to say that yes, they're different, but we don't really know why. Are they different because we're on a different treatment, or because is it the other way around? And so there's a lot of argument about cause and effect, and these questions need to be answered. And so the first questions that we've the first sign of mechanism is really these ideas of fecal transplants or microbial community transplants, that if you took somebody's microbial community and put it into somebody else, that it seems that though it it actually does change their physical state or phenotype, if you will. So what we need to understand, and this is really, again, the holy grail, is we want to know which bugs within those communities are actually at interplay with our own biology that allows us to, that causes that change. And so understanding it at that level is where the science is at. So let's get beyond the correlations and the associations, but really understand what's happening at the mechanistic level. But can we show that certain products or certain diets are beneficial to us? Surely, at a global scale, we have shown that we have shown that certain groups of organisms or a certain signature, for example, a more diverse community is known to be better than a less diverse community. Just, again, on a very high level scale, those studies have been shown, but we don't really know why. And so that's where the science is going.
That's fantastic. Well, I don't see any questions from everybody, and I have kind of one more question. I think that you touched on something that diet does affect the microbiome. So the idea for the nutritional supplement industry, I think the goal that we have as an industry is change the inside of the body so that it can change a health outcome. How far do you think we are today from seeing that causation where we are able to create dietary changes, microbiome changes that then correlate to health changes? Has there been anything done yet, and how far do you think we are from seeing that?
Well, I think we're close. I think we're quite close to starting to elucidate mechanism and function. So I just want to clarify that we already know that there is benefit to certain microbial signatures. We already know that is essentially a reality today. That, as I said, we know that a more diverse community means that you're relatively healthier than somebody with a less diverse community. That certain organisms, of course, are pathogenic. We have a lot of knowledge at the community level as well as individual, call it strains level. So we already know a lot, but in terms of, again, what is happening at the interface of a certain organism or a certain group of organisms, and what are they actually doing to us to make us healthy, that understanding is where we're going. But I don't want to confuse the question by answering that we already know there are beneficial, that there is such thing as a beneficial community, that there are certain organisms that we do want and that we need to be healthy, and that when those go away, that we're no longer in a healthy state. So we understand that relationship. So the association and correlations are absolutely strong. Now we're trying to peel that onion one more layer and trying to understand, well, how is it doing that? How is it making us healthier? How is it not? Because you can imagine then the mechanism there allows us to characterize disease better, allows us to build better therapeutics, and better products and better diagnostics. And so that's really where this is going.
That's fantastic. Well, I don't see any questions from the audience right now, but hopefully in the days ahead we'll get some questions from everybody. I'm looking forward to next week, and for those of you that participated, we are going to be talking about the benefits of the microbiome specific to the nutrition and supplement industries, and we hope to cover everything from information that has been done on things like vitamin D and proteins and a couple of other areas, so I'm really looking forward to that. Take, I really appreciate you joining us, and I look forward to doing this again next week, and we'll message everybody with the information for that. Thanks so much for joining, everyone.